All right, we've started recording, so feel free to begin whenever you'd like.
Okay, yeah, sounds good. I'll just go ahead. My name is John. I think I've met all of you at Arvo before, or most of you, but I will be there again this year. And I'm a graduate student in Vera Bonilha's lab at the Cleveland Clinic Cole Eye Institute. And my thesis project focuses on mitochondrial DNA and aging in the retina. So mitochondrial DNA is a circular genome of roughly 16.5 kb that encodes for 13 essential subunits of the electron transport chain, with 7 subunits building complex 1, 1 in complex 3, 3 in complex 4, and 2 in complex 5. In addition to coding for these proteins, the mitochondrial DNA also encodes for its own transfer RNAs and ribosomal RNAs in order to perform its own protein synthesis completely independent from the machinery that's used by the nuclear DNA. And so mitochondrial DNA is also more susceptible to oxidative and non-oxidative damage than nuclear DNA counterparts, and that's for multiple reasons. The first, mitochondrial DNA is intronless, so meaning that any sort of damage or mutation is going to occur in a coding region. And then mitochondrial DNA is also close to the production of free radicals. As we all know, the leading endogenous producer of free radicals is the electron transport chain. And then unlike nuclear DNA, mitochondrial DNA lacks protective histones. And then what's most relevant to all of us, though, is that patients with age-related retinal disorders have shown increases in mitochondrial DNA-associated damage, which has provided really a novel approach to investigating disease manifestation and how it progresses. And so perhaps the most seminal paper to investigate this idea first came from Dr. Deborah Farrington's lab back when she was still in Minnesota. She had a great collaboration with the Lions Eye Bank and was able to obtain human donor tissue specifically from the RPE macula of different stage AMD patients. And so first, looking at total mitochondrial DNA content, their group found that with age, mitochondrial DNA content is decreased in the RPE. But where things really become more interesting is when they decide to look at the amount of lesions in mitochondrial DNA. So as you can see, they found no correlation between mitochondrial DNA lesions and age. But this time, when they delineated the patients by MGS stage and looked at lesions, they found that at MGS stage 3 and 4, patients had significantly more mitochondrial DNA damage compared to stages 1 or 2. So this really provided the basis that, at least in AMD macula RPE, mitochondrial DNA damage is appearing as a phenotype of later stage AMD, possibly maybe promoting the progression of the disease. And so since Farrington's group published this work, and even maybe sometime before, this idea of mitochondrial dysfunction in the field of general retinal degeneration as a whole has kind of started to explode. And so there are groups all around the world investigating mitochondrial dysfunction as it relates to things like glaucoma, retinitis pigmentosa, diabetic retinopathy, and just a number of other retinal degenerative conditions. But what's interesting about these studies and these reviews is that they all seem to recognize that mitochondrial DNA damage is playing a contributing role to mitochondrial dysfunction in the retina. And so regardless of the recognition that mitochondrial DNA damage has received, there are still large gaps in knowledge as to how this is actually contributing to pathological phenotypes in the eye. And so one question that remains unanswered is that we know mitochondrial DNA damage leads to mitochondrial dysfunction, but what is the impact of mitochondrial DNA mutation burden in this process? So during the natural process of aging, we all accumulate mitochondrial DNA mutations in our cells. But what if this process is somehow accelerated in the retina, or it's even further accelerated in a diseased retina? And then how does this mitochondrial dysfunction ultimately lead to retinal degeneration? And what consequences does it have on retinal health? Since the retina is a post-mitotic tissue, non-renewable by new cell division, do these dysfunctional mitochondria have more impact on overall health? So these ideas are kind of the premise for where I started my thesis project. And over the last several years, I've been making these small contributions to bridge some of these gaps, with the hypothesis that the accumulation of mitochondrial DNA mutations is playing a causative role in the onset and progression of retinal degeneration. And so in order to test this hypothesis, we really needed a good model that represented mitochondrial DNA mutation burden. And so luckily for us, a mouse model already existed and it's commercially available from Jackson Labs. And this is known as the POG D257A mouse. So this mouse was created back in the early 2000s to study the effects of aging on skeletal muscle. But this mouse has a single amino acid substitution, and this is in the exonuclease domain of the POG protein. The POG protein is the polymerase responsible for mitochondrial DNA replication. But this substitution doesn't render the polymerase non-functional, but it rather makes it so it's only incapable of conducting normal mitochondrial DNA repair. So this means that mitochondrial DNA is still replicated normally, but it now has a 2,500-fold increase in mutation rate. So this mouse has already been used in several studies to investigate accelerated aging and disease phenotypes. But what's really great is that it's never been used in retinal-related studies. And so 12-month-old mice that are positive for this mutation display graying hair and a number of other general, phenotypes that represent aging. And then also, as we all know, mitochondrial DNA is solely inherited from your mother. So this kind of makes breeding these mice a little tricky. So we don't want to introduce any mutation burden at birth. We have to constantly be renewing fresh breeding cages with first-generation heterozygous animals. And so to generate these animals, we have to backbreed a heterozygous male with a C57 female in order to not introduce any mitochondrial DNA problems right from the get-go. And since we're also interested in, how aging affects, mitochondrial DNA mutation acceleration, we are studying 3, 6, and 9-month-old time points. So the first thing that when we started breeding these animals and getting mutants at their specific ages, we wanted to start just using some of the in vivo techniques that we have available to us in our department. So our first evaluation of these animals came from the eye image core we have here in Rupesh Singh, where we performed optical coherence tomography to really look at a morphological level what was happening in the mice retina. And what you can see in panel C here, by 9 months of age, compared to wild type, the POG D257A mouse in green had a significantly reduced total retinal thickness. And this reduction was further delineated into exactly what cell layer it was. And this is the outer nuclear layer of the photoreceptor cells, which actually show reduction as early as 6 months of age and further reduction at the 9-month time point. And you can also see that kind of visually here in the example OCT images.
Hey, John?
Yeah.
I'm sorry if I missed this, but do you sequence the mitochondrial genome for each of these mice?
Did we sequence? Is that what you said?
Yeah, like maybe for the retina that you're analyzing, do you ever sequence it at the end?
We have not yet sequenced it. We do have an assay in the works where we're quantifying mitochondrial DNA damage the same kind of way that the Farrington paper quantified DNA damage. But there has been a previous published work where, like, it was from a bioinformatics lab. I can't remember the exact. I should probably reference it in the background of the mouse. But they looked at several tissues. And the retina was one of them, and the brain was also liver, all sorts of different tissues. And they did see elevated mitochondrial DNA mutations in the retina as well.
Yeah, I know of that paper.
Okay.
And I guess the reason I'm asking is more than the number of mutations, it could be that the location of the mutation really matters. And so when you see some mice that have a thinner ONL very early, it might be a very critical site for maintaining, retina function or structure. So it would be good to see, because you might expect different, the number of mutations in different spots might mean different things.
Yeah, you're exactly right. And so I think that paper was talking about more so like a heterogeneous mixture of mutations, like, dependent on mouse. It's not like the dysfunctional exonucleases specifically, targeting, complex 1, for example, right? That it's not replicating or repairing complex 1 normally, or, mitochondrial DNA encoding complex 1 normally. But when we do our DNA, mitochondrial DNA damage assay, we know specifically what genes are in those regions. And so if we have a specific region that pops up with, more damage or more mutation, then uh, we can go back and look and see which one of those complexes are affected. And I'm going to show some protein data, like at the very end, actually, where we looked at mitochondrial complex proteins, and we do see a preferential decrease in some of those. And so just to kind of validate the OCT findings, we started using some histological sectioning and toluene and blue staining. We kind of see the same phenomenon, except here we don't have, nearly as many biological replicates. But there is a trend in decrease of total retinal thickness, and then there is a significant decrease in the outer nuclear layer thickness of the retina by 9 months of age. And so moving on now from the morphological data, we next performed ERGs. And so these test more of the functionality of several retinal cells. And, we also wanted to try this just to see how the physiology of the eye was being impacted. And so what you can see is that these are waveforms, I'm sure we're all aware, but each peak represents a different response. So what we saw was a decrease in A wave by 6 months of age, which is the rod photoreceptor response, and then or just general photoreceptors, and then also a decrease in B wave amplitude, and probably the most significant decrease was in the light adapted response, which is a measure of the cone photoreceptor cells. And so these are all occurring, as early as the 6-month time point, just like we saw, the decrease in the OCT as early as the 6-month time point in the ONL. So we recently submitted this paper, and we got the revisions back just maybe a few weeks ago now. And one of the reviewers pointed out that we should probably look at the relative B wave by A wave and see if the reduction in the B wave, the bipolar cell response, is just because of a reduction in the A wave. And we did that, and we actually do see that all of the points are falling basically entirely on the red line, which is meaning that the reduction in the A wave and the B wave is equivalent. And so the reduction in the B wave is not due to bipolar cell dysfunction, but it's more so just the change in phototransduction from the reduced A wave. And so that was a good point that was made, and we're going to include this in our resubmission. And then to validate the ERG findings, just like we kind of validated the OCT findings, we used IHC and probed for several well-established retinal cell markers. So the first thing that we looked at was red-green opsin, which is a marker for cone photoreceptor outer segments. And we saw a reduction as early as 6 months of age and the RG opsin signal. By 9 months of age, it looks like aging alone has reduced this enough in the wild type to make it kind of catch up to the mutant mouse. With rhodopsin and PKC alpha, rhodopsin is our rod photoreceptor marker, and then PKC alpha is the marker for bipolar cell synapses. We saw a reduction, but it was only reduced with aging. It wasn't reduced, between the genotypes at a specific age. It was just overall reduced with aging in the mutant mouse. So now that we had conducted, a pretty thorough characterization of the retina of these animals, we wanted to next take a look at the retinal pigment epithelium. And what was interesting is that when we were conducting the previous retinal stainings from the last figure, we were noticing these odd-looking autofluorescent specks at the layer of the RPE. So we decided to just kind of take a closer look at these by simply mounting a few slides and looking at them under the laser with no staining at all. This is just autofluorescence. And what we saw was that these granules, is what we're calling them, are more prevalent, or more, there's more of them present at the 3-month age time point. As you can see, the count is higher. We did like a image J thresholding. And then as the mice age, the granules start to level off with aging as far as the amount. But what happens is these granules start to accumulate and form these much larger structures. And so by 9 months of age, these structures are significantly larger in the mutant mouse. And you can also see that in the summary statistics. I just included that here, that the average granule size goes up with age and with genotype, and also the maximum size of the granule goes up with age and genotype, with the largest granule present in the mutant 9-month-old mouse being over 25 microns. So in hopes that we could figure out what the presence of these granules was from, we performed electron microscopy of wild type in the D257A RPE, and we noticed a lot of different things. Nothing that really indicated exactly what the granules could be, but we did see that the mutant mice seem to have more disorganized microvilli in the RPE. They have disordered looking pigment. It's not very apically aligned like it's supposed to be. There's the presence of these outer segments, undigested outer segments near the basal layer of the RPE that we saw. And then we also, not in every single image, but in some, we saw the presence of more of these vacuole-like structures in the RPE. And probably most significantly is this basal infolding being disrupted or disorganized looking in the mutant mouse RPE compared to the wild type. So nothing really to indicate what the granules were, but we're thinking that maybe it's something that actually gets removed during this processing for electron microscopy. Maybe something that we could pick up in like future proteomic studies or something.
John, this is Jason Miller. On the previous picture, you had your autofluorescent granule was up into the outer retina on that bottom right one.
Yeah.
How much did you see that phenomenon of autofluorescence that had escaped into the outer retina?
Oh, that was, probably maybe one of the only images that actually had that. Yeah.
Okay.
But it was just a good image to show that there is something that's very like accumulating. And I'm not sure if it was secreted or something up there, but
that wasn't a widespread phenomenon that you saw.
No, not a widespread. It was more so like, can you see my cursor, by the way?
Yeah, we can see it.
Okay. Yeah. It was more so things looking like this, where they were starting to clump together,
Thank you.
Yeah. And overall, like the minimum granule size or whatever, the mode was always around the minimum. So there's always, regardless of the time point, mostly the small granules. But in some cases, like when you took the top 10 particles per image, the D257A had the largest ones. Okay, so the next thing, we've conducted a characterization of the retina, and now we've slightly looked at the RPE. We wanted to more mechanistically investigate why we're seeing these sort of phenotypes. And so the first thing that we wanted to check out was to see if oxidative stress was playing a role. And so for this, we performed a protein carbonylation assay to detect the most common protein oxidation products in biological samples, which are carbonyl derivatives of specific amino acids. And so this assay measures 2,4-dinitrophenylhydrazine to identify protein carbonyl groups directly correlated to protein oxidation. And we were kind of surprised to see that the retina and the RPE, both tissues were used separately. These are protein lysates. Didn't show any increase in protein carbonylation with age or between genotypes. And so another specific thing that one of the reviewers pointed out when we submitted this paper was that the protein carbonylation assay is kind of a more of an outdated thing, and it doesn't specifically measure all of the forms of that oxidative stress could be coming from. And so to supplement this, we recently just did a 4-H&E stain, which is a byproduct of lipid peroxidation. And so I looked at this in 3 and 6-month-old retinas, and I quantified it literally just yesterday. I used actually these images themselves instead of a high mag RPE to try to quantify the RPE, but I do plan to go back and take a high mag 63x RPE image to get maybe a better quantification here. But again, it doesn't seem that, the 4H&E is increased significantly, at least with aging or between the genotypes of the mice.
Hey, John,
Yeah,
we've never had any luck getting 4H&E to work in any
No.
model for oxidative stress.
Yeah.
We couldn't even come up with a good positive control to see an increase, like if we treated with like intravitreal injection of terbutyl or TVH hydroperoxide or whatever, anything that could induce oxidative stress, we couldn't get a good increase in 4H&E signal.
Okay.
I don't know if it's because of fixation, like if you're using paraformaldehyde to fix, you'd be washing out a lot of the lipids.
Yeah.
So there's a lot of issues that we just haven't been able to resolve to use H&E as a good marker for oxidative stress.
Okay, yeah,
I
that
can,
that
I
can
can
make
second
sense.
that for in vitro. We can pour whatever oxidant you want on our cultures and not get an increase in 4H&E stain by Western or by ICC.
Okay. We actually, I did this just to try out the antibody because we ordered it, and then I wanted to test it before I actually used my, big important samples. And so I used an old slide that we had injected sodium iodate. It was actually a DJ1 mouse, but same thing. We injected the 20 milligram per kilogram sodium iodate, and you could actually see a much higher staining, like a baseline staining in the ganglion cell layer, and it looked almost like it was kind of infiltrating. The staining was much more significantly, brighter, I guess you could say, in those sections compared to these. So we kind of did use a positive control, at least when we were testing to see if the antibody worked. But yeah, these didn't have any sort of positive control. I just used a secondary only negative control to make sure that this was real. And then another thing that we just recently looked at, because we're trying to, expand on oxidative stress a little bit more. That was kind of one of the things that came up in the reviews. And so we probed for DJ1, which is an antioxidant protein that's used in the cells and has been shown by our lab to quench, oxidative stress, free radicals, things like that occur with aging. And we didn't see any increase in expression of DJ1 in the 3 and the 6-month-old wild type in D257A retina, at least. We haven't probed for this in the RPE yet. I'm still waiting to do those lysates as well. So, however, the one thing that we did see at least a significant increase in was when we started staining our retinas with 8-OHDG, which is a chemical modification to nuclear or mitochondrial DNA that indicates oxidative damage. And so we first tested this on the retinal sections, and we saw just a significant increase in the wild type animals with aging alone. This increase was not significant, but it was elevated in the 3-month-old D257A mice, specifically elevated in the ganglion cell layer, as you can see. And then we looked right at the RPE layer with a zoomed in magnification image, and we saw again that this 8-OHDG increased with age, but it wasn't specific to the genotype of the animal. So this kind of validates the more recent but not as well characterized idea that mitochondrial DNA mutation burden doesn't actually lead to the vicious cycle of oxidative stress. It's likely that dysfunctional proteins that encode the electron transport chain probably lead to less ATP production and therefore less endogenous free radical production. And so that's more of a new theory that's in the field of oxidative stress biology that kind of goes against the vicious cycle theory. And And so to finish up the paper, we, of course, wanted to look specifically at the mitochondrial phenotype in the retina and RPE. And so first looking at just retina isolated proteins, we performed Western blot for several mitochondrial markers. So of course, the first thing we wanted to do was see if POG expression was actually changed at all due to the exonuclease mutation. And we actually didn't see any significant change in POG expression between 3 and 6 month old animals. Which was great because we didn't really want, you know, the phenotypes that we're seeing to be because there's just lower expression of POG. We want it to be that POG is expressed. It's just the mitochondrial DNA is mutated. And so we then looked at PDHA1, which is a commonly used mitochondrial matrix marker, and VDAC, which is a mitochondrial outer membrane marker. And we saw that PDHA1 was significantly increased in the D257A animals at 6 months of age, whereas it was decreased between the 3 month old wild type and the 6 month old wild type. And VDAC was significantly increased at the 3 month time point between the wild type and D257A mice. And then the final thing that we looked at in the retina, at least, was the complex proteins themselves. And so we used this total OXPHOS antibody from AbCam, which probes for a specific subunit of each of the complexes of the electron transport chain. And we saw a significant reduction in complex 4 between genotypes at the 3 month time point, and also a significant reduction just with aging alone in complex 4. In complex 1, though, we saw a significant reduction at both the 3 and the 6 month old time point, with no significant reduction just with aging alone. And so this is really great because if you remember back to the background slides where I was going over the encoded mitochondrial subunits, this complex 1 has the most encoded mitochondrial subunits with 7 being encoded by mitochondrial DNA. And then complex 4 has the second most with 3 being encoded by mitochondrial DNA. And actually this COX-4 antibody in this cocktail is specifically targets this subunit COX-1 of complex 4. And so it's not really surprising that these are the complexes that seem to be the most affected because these are both, you know, the most mitochondrial DNA encoded. So now turning to our actual RPE protein lysates, we looked at the same exact things. POG was again not increased or decreased with aging or genotype. In this, however, PDHA1 and VDAC both were not significantly changed, unlike the retina where we did see significant changes in both of those mitochondrial membrane markers. And since we had already taken some EM photos just to try to identify what those granules are, we also went back to those and took a higher mag 30,000x image to look at the mitochondrial morphology between the wild type and D257A. And what we noticed was that the mitochondria in the D257A mice seem to be, first of all, significantly shorter in length. And they also have, we haven't quantified this yet, but we plan to, these more less electron dense mitochondria where you can actually see like more dilated looking cristae compared to like the wild type that has a much cleaner and tighter looking cristae. And then again, looking at the OXPHOS proteins and now in the RPE protein lysate, we saw a significant decrease again in the 3 month old complex 4 expression in the D257A, and also a significant decrease again in just aging alone with complex 4. Complex 3, we saw a significant decrease with aging in the wild type, as well as complex 5 with aging in the D257A. But again, we saw a significant decrease at the 6 month time point only this time, not in the 3 and 6 month of complex 1 expression. And so that kind of summarizes the paper. I do have some more stuff I want to get into after this, but we saw both morphological and functional characterizations showed deficits by 9 months of age in the POG D257A mice. That was specifically the decrease in the ONL, the total retinal thickness, and then the ERG, A wave, and the light adapted response were reduced. And then histological and immunohistochemical staining validated these findings. We also saw, decreased ONL thickness by histology and reduced RG-opsin staining and rhodopsin and PKC alpha staining. And then the RPE of the POG D257A showed increased autofluorescent granule formation and accumulation. And then the EM showed, just those general changes I talked about in the RPE cell and mitochondrial morphology, where we saw reduced length of mitochondria and misaligned pigment granules, disorganized basal enfoldings and microvilli, things like that. And then surprisingly, we didn't see that oxidative stress is accelerated in the D257A retina or RPE. This again maybe goes against the well-studied vicious cycle theory of oxygen or reactive oxygen species production. And then mitochondrial proteins in the D257A showed different expression when compared to the age-matched wild-type animals. Specifically, the strongest data suggesting that complex 4 and complex 1 are the most compromised. Does anyone have any questions on that? I want to go into, so I have a lot of current work in progress too that I wanted to get into, but if you have any questions on this is like the paper itself.
I have a comment about the oxidative stress idea or results, I guess.
Mm-hmm.
And I think one thing is a lot of people measure, like one of the major antioxidants, at least a lot of people consider it a major antioxidant, is glutathione. And glutathione is constantly regenerated by NADPH. So that's reducing power that you spend to sort of keep your antioxidant system alive. And I guess one thought I was having for your talk was, if complex 1 is down early, then perhaps you have a surplus of reducing power that could instead be used to regenerate glutathione rather than generate ATP. So in a world, it's kind of a long sequence of events, but there could be a world in which it makes sense that you're not seeing more oxidative stress due to a decrease in complex 1 activity.
Oh, okay. I've never thought about it like that. Yeah. I was just thinking of it more in general, like the ATP production itself produces, oxidative radicals. And if you don't have expression.
Yeah, I think more of the mitochondrial reactive oxygen species get produced when there's an imbalance, like say less complex 4, but not less complex 1, and then you have like a sort of backflow of electrons.
Yeah. Something that we do want to do that we haven't really gotten into yet, but we do have plans, is like an activity assay on these complexes, and also look more specifically at like the super complex formation, because these don't really exist exactly how they're shown here. They exist more as, like a bundle of complex proteins. And so that's called a super complex. So maybe we would see decreases in that. I'm not sure. Something we have in the pipeline though. But yeah, I think that there's a lab here that has a good GS antibody for Western. I might take a look at that and see if we see different expression. Would that kind of indicate, I guess, what you're trying to say, Dan?
I think if I was looking for glutathione redox status, and I'm not sure that that's the thing that you should look for, then I would use, they have assay kits, for example, that you can get from, I don't know, Cayman or Sigma or something, one of those companies, that you'd use a plate reader and homogene it. Or you could use mass spec, but yeah, I don't know if an antibody-based approach would help with this particular marker. But I don't know if I would recommend you sort of beating a dead horse when it comes to oxidative stress, because you already have this really interesting result in the complex 1 and complex 4 levels.
Yeah. Okay.
I was just gonna make maybe 2 comments. I think it's really, well, I guess first off, have you taken eye cups and figured out what the OCAR activities are at different, like what's the functional
Yeah,
damage in terms of
that,
metabolism at different time points? Like, are we dealing with a highly inefficient mitochondria,
yeah.
or is somehow there's a lot of compensation? Um,
The
because
current work,
yeah.
includes some seahorse data that I'm excited to show you actually. So,
Okay, because I
yeah.
think that leads to the second point. At least Dan and I have, and Jim have talked about this. It's not clear to me whether or not the RPE is in general limited by the need to produce energy or the need to produce a bunch of other things. For example, Dan talking about the antioxidant defense status or the redox status or a range of other things. Because of
Mhm.
course the RPE is sitting next to a capillary bed that has one of the highest flow rates in the whole body. And so it's, even though there's a lot of metabolic activity in the outer retina, I don't know, some of your results, the fact that actually the the degeneration is not entirely, immediate and rapid, the fact that,
Mhm.
that actually I'm surprised at how well things function. If you then show that the mitochondrial status is highly compromised, then it may make an argument that the RPE is not really energy limited. It's other processes that the RPE is using metabolites for. If
Yeah.
instead you see that despite all the mitochondrial DNA mutation, you have good OCAR, then that suggests the RPE has, of course, a resilient capacity to maintain mitochondrial capacity. And so I think that could be an interesting sort of diversion between two, or diverting point between two hypotheses.
Mhm. Okay. So some of the current work that I have going on, after, we've submitted this paper, is I'm still continuing, in the animal model itself. Some things I'm interested in looking at are like glucose and lactate transporter abundance, like GLUT1 and the MCTs, probably by staining. And then we are currently conducting some seahorse assays looking at oxygen consumption and extracellular acidification. And then, like I mentioned before, we do want to isolate DNA from these animals and look at exactly, how damaged the D257A is over the wild type with the time points that we looked at. But something that I really don't have time to talk about today, and I have this is like a whole other project, is I also have a cell model of mitochondrial dysfunction where I've knocked down polymerase gamma, and I'm going to overexpress it in various human cell models. So uh, the blot I'm showing here is actually an ARPD19 model where I knocked down polymerase gamma. And then with this, we also have plans of doing oxygen consumption assays using the Recipher we have, and then some live cell mitochondrial analysis and a bunch of other things, but those are probably the key ones. And so the last kind of couple data things I want to talk about are, we have started doing some retinal explant, or not explant, but retinal punches, seahorse assays, using a mitochondrial uncoupler and a respiration blocker, and looking at specifically young and old retina now we're calling. So the young is like the 3-month time point, and the old is not the 9-month time point, but we actually extended this even further to a 12-month time point. By about 13 months of age, these mice are um, pretty much dead. I mean, there's not many that live much longer than 13 months of age, the D257A at least. And so the 12-month-old is kind of like, a very old time point for them. And we do see a significant reduction, at least from the preliminary, punches that we've taken and assayed, in the old animals' basal respiration, and also a significant reduction in their maximal respiration at both young and old time points. What's interesting, though, is that the respiratory capacity of the old mice is actually significantly increased compared to the wild type. So this could indicate that, it's likely that they have the capacity to perform, oxidative phosphorylation, but they, the disruption of the electron transport chain proteins or something like that is making it so they actually cannot consume the oxygen. And so another thing that we looked at was just the ECAR, which is kind of a measure of like the glycolytic consumption of the animals. And at young, we don't see a significant decrease or increase in resting ECAR. But what's interesting is that the old animals seem to have less resting glycolysis when compared to the young. Although this isn't like significant between the two genotypes themselves, it's just significant with age. And then the last thing I wanted to talk about was just kind of some plans. Maybe I could get some suggestions here. We have this idea of, we've already created this cellular model where we've knocked down polymerase gamma, but we're also wanting to, we've bought Lenti and AAV viruses that have full-length POG and this mutated, this is the human homologous POG called D274A. That's the same exonuclease domain mutation as the mouse model. And then, also some empty virus to use on maybe a negative control cell line, like ethidium bromide treated, where the mitochondrial DNA is completely depleted. So that hopefully we can, kind of more mechanistically look at the stuff that we looked at in the animal model, but just a little bit more deeper and more insight and things like that. And so those are my plans with the cell model. And with that, I just wanted to thank everyone that's helped me along the way with this project. I had a lot of help from my lab and then some other people in the department, specifically Shiju and Coco. Shiju helped me with some of the Western blots and Coco helped me with a lot of the seahorse data. My committee and then all of the cores that have helped me. And there's my contact information if you guys want to reach out about anything. But thank you for listening.
Eric?
Any more questions maybe about the seahorse or anything?
That was really interesting work, John. Thank you.
Yeah.
I guess a comment about your animal model. Have you done any target metabolomics or anything, or have you looked at NADH or NAD plus levels in those animals?
No, we haven't. It's something that we want to do. Like we are collecting, like, as we get animals at different time points, retina and RPE, to perform both proteomics and metabolomics. I think we're going to write maybe like a little core grant to try to get some, small funding for those experiments and things like that.
Yeah, it kind of reminds me of our GOT1 knockout just a little bit.
Yeah.
Because we have an accumulation of NADH that might be happening if your complex 1 is down. Like Dan said, it could be causing this reductive stress. So it'd just be something interesting to see if you're just getting too much NADH or it's disrupting amino acid transport and usage also. Like we look at ISR or the integrated stress response, if they just can't, keep manufacturing outer segments because they can't maintain their, not an essential amino acid homeostasis, that would be why you're getting such a slow degeneration.
Yeah.
And then for your RPE, your ARP19 work, wondering why you want to knock out Paul G instead of doing something like complex 1 knockdown. Because I assume like when you knock down Paul G, you're gonna have to wait a very long time for mitochondrial DNA mutations to occur. And if you think it's like the complexes, why not just go right for them and see how they're affecting your RPE health?
Yeah, I think the idea with like the POG knockdown is that, we want to overexpress this this polymerase in those cells to try to get them to use this rather than the lower endogenous POG, I guess. And so the knockdown itself wouldn't create mutations, but this usage of this polymerase would. You get what I mean?
Right, but it could take
forever
to generate enough mutations to get a phenotype like you've seen in your animal model.
Yeah, true. You're right. using this, and then isolating the cells in 2 days, you probably wouldn't see anything. I get your point. Yeah.
But yeah, great work. Thanks, John.
Yeah. Yeah.
John,
Yeah.
No,
other
I meant Nate.
Oh,
So John, you showed in the first part of your talk that the neuroretina degenerates over time. So I was just wondering how you normalized your seahorse data, if you took that degeneration into account, things like that.
yeah. This is actually non-normalized because we don't really have enough. These are technical replicates, like just the number of punches. I think we've only actually done like 2 mice for each time point. That's why it's kind of in the current work section. But these are like five punches each mice or whatever. So that's why you get around 10 dots. But we have taken an extra punch or 2 from each of the animals that we've used for this assay, and we were going to perform like a micro BCA on that and check like maybe protein expression and see if they're different, and then normalize to that protein expression. Or yeah, just total protein, I guess is what I would call it. Like
Yeah.
OCR divided by total protein.
Mm-hmm. Okay.
Yeah. I know that like in the past people have shown like total DNA content, because when you're working with a punch of retina, it's very hard to get a lot of protein in the first place. But if you're starting with the same size material,
Yeah. No,
but you could be right. These changes might not actually be real once we do that step. And I think that I don't really want to isolate the protein from those little punches until I have all of them collected and do them all at the same time, just so there's no like variation between running it one day versus the next.
Okay.
Yeah.
Okay, that sounds good. Thanks, man.
all right, Jim, your hand is up.
Yeah.
Oh, yeah. I had a general question. It's just, with all this, you have a favorite? So the ERGs went down, and I must, maybe I missed. I was walking to work, but were the cells dying? Are the cells dying? Is there apoptosis?
I think for sure by 9 months of age, there's definitely cell death in the ONL, because the outer nuclear layer looks not only thinner, but it looks like there's less nuclei. So, I mean, this is not something we've quantified, but we could probably easily count the number of nuclei.
Yeah, that'd be interesting to do. But I just wonder, like, what, with all the work that you've done and everything you've seen, do you have a sort of favorite hypothesis for why they would be dying, or is it lack of ATP, or are there any new insights on photoreceptor cell death you might be getting from these experiments?
I mean, I think my favorite result that probably took the longest was the complex result. And so when I got this block, it's this is one of those things that really makes you happy, right? So when you see something like this. But yeah, in general, probably lack of ATP production, or maybe like altering one of the levels of these complexes probably alters the super complex formation as a whole, which we haven't really looked at yet. But that could be the reason why we're seeing retinal cell death, or outer nuclear layer thinning and things like that. But again, it's weird because the retina, is not supposed to really rely on mitochondria and oxidative phosphorylation. It's supposed to be receiving glucose from the RPE, and that whole ecosystem, right? But it does seem that they do rely on mitochondria and oxidative phosphorylation. If we're seeing that deficits in those result in likely the cell death of rods or cones.
And is it possible, the way that you do the experiments, to measure the ATP levels in the retina and the RPE? And the retina seems like Jason was sort of saying before, maybe the retina has more energy demand, so maybe with, given the same mitochondrial defects, the retina might be more, its ATP levels might be more sensitive to, to all these mitochondrial mutations than the RP would. Have you measured the ATP levels, or
No,
maybe I
no,
missed that too?
we haven't looked exactly. No, but again, it's something we want to do. Yeah.
Yeah, thanks.
Okay, so I guess my question is a little bit more technical, and it's good that you brought up this figure because at the 3-month stage, you see like 50%-ish loss, maybe more of complex 1 and 4. Complex 4 is the thing that actually consumes the oxygen. And this is one subunit, so that's, I guess, worth noting.
Yeah.
And so the question is, why don't you see in the young OCR data a 50% reduction in respiration?
Yeah, that's odd, right? That yeah, it's the same time point, and we don't see the reduction like in the OCAR units at that point. But I don't know. I haven't really thought about it, but maybe there's some sort of compensation that's occurring at the young age that's unable to be compensating as the mouse ages and accumulates more mutations. Do you know what I mean?
Well, you can't, I don't think there's anything else that consumes oxygen as well as complex 4.
Yeah.
So like the question would be, what's compensating? I think Jason's point kind of comes up here of, maybe like they're not working, they're not working their complex 4 as much as they possibly could. And so even a reduction in complex 4 is tolerated by the system, because OCR doesn't need to use all of the complex 4s that are there. But I usually consider that to be, I mean, in the light of all of the seahorse and non-seahorse OCR data with retinas, where they show, treating with FCCP results in no gigantic change in respiration, it suggests that, retinas are working at maximum capacity. So that kind of contradicts this notion that, and it suggests that you should see a 50% reduction in respiration.
Yeah.
So I think this is sort of a general issue of, why don't those two things match? And I don't have an answer, of course, but it is.
Yeah, the only thing I could also say is that this is actually an antibody specifically for this COX-1 of complex 4. So it could be that the complex 4 as a whole is still functional, and that's why the OCR data is still okay at this age. And then the actual protein expression itself is reduced because this is the mitochondrial DNA encoded subunit, right?
Yeah, so I guess it is definitely worth doing what you were suggesting earlier and measuring the activity, the enzymatic activity of complex 1 and 4.
Yeah.
All right. Jim's hand is still up, and I don't know if that's a new question or just from before. But if not, then Sai, please go ahead.
Hey, John. Thanks for the great data. I really love your talk. Well, what I noticed was that in the RPE ultrastructure data, I noticed what you called as vacuoles were they actually look like lipid droplets to me. So the thing is, would you expect, or I mean, with your data or with your mutations, would you expect there would be lipid accumulations? And if so, I mean, mitochondria has to oxidize lipids, but then you're only affecting the OXPHOS protein. So can you see any way that the lipid accumulation would be occurring, or like lipid oxidation is affected in these cells?
Yeah, I think for sure that lipids could be affected as well. It's just not something we were really focused on. But again, like we weren't seeing these structures in like every single section. We performed 3 biological replicates, and we just saw them here and there kind of thing.
Right.
But
no, it was just interesting to see this happening in the mitochondrial problem.
Yeah,
RP
in a
as well.
model. Yeah.
Thank you.
I would say like the most pronounced thing that we saw in like every single image that we're taking is this difference in basal enfolding organization, which we didn't really know how to interpret, but these again were to try to find out what the the granules themselves that we showed were actually if we could actually see them by EM, and then we just noticed all these other little things here and there.
Yeah.
Mhm.
Well, the basal enfoldings and the microvilli problems could mean various things, but one of them is also that the RPE might be dedifferentiating, or it may be either be a developmental problem or a degenerating problem where RPE is dedifferentiating. But that's probably something that is not very something you're looking at. So
Yeah.
Thanks a lot.
I'd just like to follow up. So, John, this is a great presentation. Steve Leisler here. So to make a compelling case about the basal enfolding, you'd have to do a lot more sampling and do a lot more quantitative image analysis to make it compelling. But we see this kind of variation from one part of the retina to another in the same eye. So you just have to be very careful about those kinds of
If we're in like the same regions and things like that.
exactly.
Okay.
Yeah.
One thing that I also noticed about these images, and we don't really know what they are, but do you see how the pigment in the D257A has these odd, like almost looks like a hole right next to it, right, in the tissue, whereas these are much more like they look better, I guess is what I could say. We were thinking maybe that this could be what's contributing to that granule that we see in the autofluorescence study, but not sure.
The other thing is that in that right hand micrograph that you're showing with the asterisk,
Yeah.
that it's very pale, which means it's not osmophilic. Yeah, that little asterisk down in the lower
This
right.
here?
Yep, right
Yeah.
there, right there. And so possibly that whatever is in there that likely is a lipid deposit is probably not enriched in polyunsaturates. Saturates don't stain very well.
Okay.
Okay, so one last thing is that we got from Nankai Wang, paper posted about protocols to measure mitochondrial function in tissue homogenates. And, I think function probably has to be interpreted pretty broadly, but it looks really neat.
Okay.
So it might be a useful resource for you. And I could send this to you after if you don't want to take the time to copy paste it, but it might be a tool. Okay, thank you everyone for your questions. I hope to see many of you, if not all of you, at ARVO. And we have the happy hour thingamajig, so please show up if you want. And I'll be sure to find a location. And thank you all.
Thanks, everyone.
See everybody.
Ciao.
This is like the chance to see who left their computer or phone in some cases.
People must know we have our secret meetings after.